Welcome, everybody. I'm Mike Saag from the University of Alabama at Birmingham. And today with me is Dr. Mark Mulligan, who's the Division Director of Infectious Diseases at NYU and Langone Medical Center in New York. And today's topic is COVID vaccines. This is being brought to you by the IAS-USA, the International Antiviral Society USA, based in San Francisco. This is one of the continuing in the series of the COVID dialogues. Couple points about today's session. It's going to be about an hour long. It'll be conversational between Dr. Mulligan and me. Our goals is to cover all things COVID vaccine. Mark and I will speak together for about 35 minutes, and then we'll open it up for questions and have a round table. If you have questions, you'll notice at the bottom part of the Zoom screen that there's a Q&A box. Just submit the question there, and we'll get to as many of those as we can when the time comes. As a special note, the chat feature has been disabled so that we are just using the Q&A boxes for dialogue to ask the questions, and that keeps everything in one location. An important note, these dialogues I think are very informative, but they're not CME accredited, so there won't be any CME available for the dialogue itself, but there will be web-based and podcast broadcast available that can be available for CME, but with the warning that the shelf life, if you will, of COVID information goes stale pretty quickly. But I think we're going to cover a lot of basic points that'll be real helpful. So I think without any further ado, we will get started. Dr. Mulligan, welcome.
Thank you, Dr. Saag. It is really great to be here, especially with you, my colleague and friend.
Yeah, we go back a long way, longer than we'd like to maybe remember. But anyway, let's dive into this.
Okay.
I'm gonna divide this up into a couple topic areas that I think we can roll through pretty quickly. But the first will be about science, and then timing of vaccination and safety, and finally immunity. And then maybe if we have time, we'll get to issues about distribution, trust, mistrust, and that type of thing. So let's go. How do these two vaccines that have been approved work?
Yeah, it's called messenger RNA, and they're in these lipid nanoparticles. So you can think of copies of RNA of the spike gene that get delivered intramuscularly right there in the deltoid, get taken up into muscle, but also probably importantly into professional antigen presenting cells and go to regional lymph nodes. So, we do see some regional node swelling in some of the vaccinees, not many, but some. And there you get induction of an immune response. So the RNA goes into the cytoplasm. These lipid nanoparticles get taken up by the cells. They're about the size of a virus. They're 50, 80 nanometers. HIV is about 100 nanometers diameter. And once they go inside, the RNA is released into the cytosol, the cytoplasm. Very important. Never goes into the nucleus. Gets degraded within less than a day or so. And the RNA, messenger RNA, messages the cells. It tells the cells, hey, make this protein, make the spike. And then the spike protein is made. It's anchored. It's a trimer, very importantly anchored in the membrane. It has two proline mutations that stabilize the pre-fusion configuration, locks it in. Very important information that was available, when this vaccine was designed based on earlier work that had been done with MERS and with RSV. And then that endogenous, this is the key, that endogenous synthesis of the vaccine protein right in the body, asking the body to be the factory to make the virus protein, the spike vaccine protein. You get a more natural immune response, balanced. You get CD4, CD8, B cell, antibody, and a very potent response.
So when you're saying a couple quick follow-ups, when you say cells, you mean the muscle cells?
Well, yeah, I think muscle cells are part of it, but also, Mike, I do think that there's uptake into the macrophages and to dendritic cells that then travel to the regional lymph nodes. And in that very rich milieu, you get presentation to T cells and B cells and get a strong response.
Yeah. Any adjuvant?
These are considered self-adjuvanting. RNA binds to TLR7 and 8 and trigger an innate response. These RNAs have been tinkered with a bit. There are a lot of changes to the sequence that are synonymous, but that reduce a little bit that TLR7/8 triggering. But there still is, perhaps through the lipid nanoparticles and what they're doing, there's still a fair amount of triggering of an innate response. So there's no adjuvant, but they're self-adjuvanting.
And that probably relates to safety, I would imagine, because some of the reactions of other vaccines are to the thimerosal or the other adjuvants that are used to stimulate the immune system, right?
That is true, yeah. The tolerability of the vaccines relates to the strength of often of that immediate innate adjuvant induced response. And sometimes that can be too strong. But in this case, I think we tried to pick it, the middle ground, the sweet spot between tolerability and induction of a strong neutralizing antibody response. And I think for each vaccine, it's interesting, the two vaccines, one of them is using a 30 microgram level dose of RNA, the other uses 100 micrograms, but that's what worked. And they're a little different in terms of the lipid coating of the RNA. So it's what works, and it's what's tolerated, and induces obviously a very effective response.
So these lipid nanoparticles don't require a receptor, they just fuse with the cell membrane, right?
They get taken up through endocytosis and fuse with membranes and release the contents, the RNA, into the cytoplasm.
Okay. And we've seen the data, I think most of us, but just briefly, what it looked like in looking at the curves is that in the placebo group in both studies, and you were leading the Pfizer study, that the placebo group just kind of had a trajectory. It was pretty linear. It just kind of went up. But the vaccine group continued in the same path till about day 12 or 13, and then it just flattened out. There were some cases, it seems like, that happened after day 13. This is after the first dose, before the booster, right? And it just stayed flat. What did you think when you saw those results for the first time?
I just thought that this is truly amazing, more than we hoped for. It was very exciting to look at those curves separate right there where you said around day 12, 13. And I think that I've heard Dr. Fauci say, hey, we'd have been very happy with 70% to get 95% protection. Oh my gosh, amazing.
I remember back in the day with HIV when we were working with viral load, and you were at UAB at the time. I think you were probably a fellow, and or maybe just on faculty. And we started working with viral load, quantitative viral load, for the first time. And when those data came out, I'll never forget that moment. It's almost like it was made up in a dry lab somewhere, and just almost too good to be true, right?
Yeah. I've heard it said, and I've only had this experience a few times in my career, that science is really a few moments of sheer exhilaration that punctuate an awful lot of hard work. Yeah. And this was one of those moments, and your moment there was another.
Yeah, well, let's take that 12-day, 13-day issue, and Bob Wachter and a colleague from Harvard wrote an editorial in Washington Post this weekend about the notion of prolonging or waiting for the second dose, and with the notion you could maybe wait 2 or 3 months because that initial benefit starts to accrue at day 12 or 13. And their theory is, and they're probably right, I would guess, is that the second dose is really mostly for a booster effect or longevity, and maybe not so critical for protection maybe in the first couple months. What's your opinion on that?
Yeah, I think that what we see at day 12 or 13 reflects what happened 6 days earlier. So really, probably, because remember the incubation period. So you're starting to protect, probably, it's pretty amazing, a week after vaccination. So very, very interesting that that's happening. And the mechanism of that, I'm not sure. It seems a bit early for an adaptive response. Is there something innate going on there that's providing early protection? Then the adaptive, the antibody T cell kicks in. But in any event, for Pfizer, it's about 50% up through day 21 when the second dose was given. And for Moderna, it's about 80, it's 52% Pfizer, about 80 for Moderna. The data are cut a little differently. I mean, they're not exact same analyses. So I think you shouldn't really compare 52 and 80, but let's just say somewhere 50 to 80%, one shot providing protection for at least out through day 21 or day 28, the second dose.
Right. So coming back to the question, then how important is it for that second dose to happen within a month, or could it be delayed without much loss of efficacy? I know it's hypothetical, but do you have
Right.
a guess?
Yeah. My guess is that, we get to partial protection with one dose. You got to have the second dose to get to that 95%. And I think you're right. Oftentimes the boost not only pushes you to a higher level of efficacy, but also gives you better duration. We don't have any information. I think that, what was studied, Mike, and what was licensed or what was granted an EUA, which is not the same as licensure, was the 2-dose regimen here in the US. And I think that, the UK, they actually have some data that support longer intervals with the Astrazeneca vaccine. They extended that to their Pfizer vaccine, which there really aren't any data for. So we just don't have the data for Pfizer. Probably it would work. It's a strategy that I don't think we're going to adopt in the US. I've heard some of our leaders like Dr. Fauci say, no, we're sticking with what we know works. And I think probably that's the right thing.
Yeah, I kind of agree with you in the sense that our problem right now is more logistics and getting it out and getting it into people's arms. And if we really focus on that and make that happen, then I think we can probably keep the schedule as it is. Switching over, you mentioned AstraZeneca briefly. Describe that vaccine and how is it different?
Sure. So we've got multiple platforms. We've been talking about RNA, Pfizer, Moderna. So now let's talk about non-replicating viral vector, adenovirus. Interestingly, this is a chimp ad. So it's different than the ad5 that a lot of the HIV folks remember that we used it for HIV years ago. So it's a serotype of adeno that few humans have ever seen, and there's little cross-reactivity to. 2-dose regimen, a month apart, and phase 3 trial ongoing. We're participating. Got about another week of enrollment in the US. We're over 30,000 enrolled. We're finishing up with some enrollment to try to increase the numbers of minority participants. So we're going to over-enroll a bit for a week or two. But it's a vaccine that we know is efficacious from data in Brazil and UK. And in fact, I think everybody on the call probably knows last week, UK licensed it or made it available through an EUA again, or whatever their equivalent of the EUA is. So it's now being deployed this week as an approved vaccine in UK. The protection level depends which data set you look at. It's between 62 and 90%. If you average it out, roughly 70%. We have a bit more to learn. There were some unique aspects of the trials that were done in UK, and our US trial will be done, with a very high level NIH quality, rigorous work, and we're going to get to the truth about where it is. But I am confident that it is an effective vaccine, and will almost certainly be achieve its EUA probably in February.
Yeah, and you're thinking your best guess is maybe closer to the 90% efficacy if you had to put a nickel down?
My guess, it's hard to say. If I had to put a nickel down, Mike, let's just throw out 80-ish.
All right, okay. Not going to hold you to it. And what about safety for AstraZeneca?
This is on people's minds. So we have to rewind a bit and remember these recombinant viral vectors have been around, non-replicating. They don't multiply in the body. It's a common cold virus. Been around for years, decades, outstanding safety profiles. And there was a pause in the US trial for nearly 2 months based on some neurologic unexpected adverse events in UK, transverse myelitis in one case. And it's not clear whether that's related to the vaccine or not. It's kind of one of those things if you don't want to throw out the baby with the bathwater. We've got a vaccine that's effective. Life happens during vaccine trials. When you enroll 40,000, 45,000 people in the Pfizer trial, people are going to have strokes. A few people are going to develop autoimmune conditions, et cetera. And you really have to look for patterns, biological plausibility, relationship to vaccine, et cetera. And to answer your question, I think it's a safe vaccine, and we don't have any reason to not think that at this point.
And real quickly, just to fill out the blank, I'm going to move on to J&J in a second, but that this is a chimp adeno, and the concern with the human adeno is that some of us who have had that adeno before, if we had had it, then the immunity sometimes isn't quite as robust when you're responding to it gets knocked off too quick, right? We saw
Exactly.
that
Yeah.
in HIV trials.
Right. We call that pre-existing immunity. It dampens the take of the vaccination. You don't get a good take because
Yeah.
you knock it down.
Which is why we're using the chimp in this one
Yeah.
for AstraZeneca. And then finally, the J&J vaccine, I understand that's a one dose, one shot
Yeah,
approach.
it is. They're studying a 2-dose regimen as well elsewhere in the world, but they've now got some 45,000 people enrolled. They've closed enrollment. That will likely be the next one, Mike, to come to an EUA, perhaps in late January, early February. So they're fully enrolled, single dose regimen, again, non-replicating common cold virus, adenovirus, again, a relatively rare serotype, in this case, Ad26. So we get away from that problem of pre-existing immunity. They are looking at a 2-dose regimen.
That's a human Ad26.
That one's a human one, yeah.
Right. Okay.
But it is a fairly rare one that very relatively few of us have any cross-reactivity or reactivity to. So yeah, fully accrued and now in follow-up. Remember to go for an EUA, there are 3 things you have to have. You have to have a median of 2 months of follow-up post-second dose for safety. You have to have your efficacy data have met the numbers needed there, roughly 150 infections, and you have to have the manufacturing data. They're going to get there probably late this month and put their package together.
Great. Let's move on. Just a couple of common questions that I'm hearing about who should get it and when. I think we've seen the rollout. That seems pretty reasonable that the healthcare workers and nursing home folks and other high-risk folks get it first, and then it'll go to the older population and onward. Some criticism has been about HIV-infected patients not being included in studies. Is that conquered now? And would you recommend this vaccine when it becomes available for people with HIV?
Yeah, it's unfortunate that we did have participation, but in the case of Pfizer, it was through an amendment. They weren't involved from the beginning. In AstraZeneca, for example, they are involved from the beginning. So well-controlled HIV, you can participate in these phase 3 trials. We had about 150 to 200 in each of the trials for Pfizer and Moderna.
And they tracked like the other participants.
Yeah, but the numbers are so small, you can't break them out and say anything in terms of efficacy. But for example, in the Moderna trial, there was 1 infection out of their 170 roughly HIV participants. At the time the data cut was made to go to the EUA, it was in the placebo group.
Right.
So 100% efficacy, but of course you can't.
Small numbers.
Yeah, you can't say anything. And then, here's where we are. It's a very safe vaccine. It's not a live viral vaccine, the kind that is sometimes not given to HIV infected individuals who are significantly immunosuppressed, well under 200, or maybe to pregnant women. This is a very safe vaccine. So we don't have data about safety and efficacy in large numbers. We know in a few hundred HIV patients, very safe, no problem. And those are well controlled. So probably generally CD4 200 and higher virus controlled on stable therapy. But I don't think there's a reason to think there'd be a safety issue. I think it's very reasonable. And this is what CDC has said. For HIV patients, they've been studied if they're well controlled, and even for those not well controlled, for immunosuppressed individuals, what's being recommended by CDC and others is that there's no reason to think there would be a particular problem. Just to understand, we don't yet have the data, but it can be given. We don't yet have the data for safety and efficacy in those groups. That'll probably come soon in follow-on studies.
And in the interest of time, I'll just list a bunch of other categories of individuals that I think also this applies to, because you nicely just described the difference between a live vaccine and for example, an mRNA vaccine, very different in terms of risk. But pregnant women, breastfeeding women, people who have got other kind of immunocompromised disorders. In my view, which is worse, the unknown hypothetical risk of a reaction versus getting COVID? And especially people we're noticing who are on CD20 inhibitors like rituximab, those folks are not doing well with the disease, and be much better to try to protect them if we can. How well the vaccine works in that setting with B cell inhibition remains to be seen, but I can't imagine that that is going to be worse than the infection itself.
Yeah, again, it's theoretical. Any side effect or risk. We excluded people with immunosuppression from the trials. We did have a couple hundred pregnancies in the trials during the trial, and those people will be followed. The guidance is that a pregnant person or a breastfeeding person can make the decision that they want to make. It's okay. They can take the vaccine. We know that there's higher risk for complications in pregnant people and for their fetuses. So it's a very reasonable thing.
Who get COVID?
Yeah, with COVID. Sorry, with COVID. Higher risk with COVID. And the same answer for those other immunosuppressed groups you're talking about, people on the CD20 inhibitors and other immunosuppressives. We don't yet have the data. And when there's not evidence, we go to, well, what's the expert opinion? What's likely? What's the risk? You balance the risk and the benefit, and that's where you started. The risk is higher in these individuals, and so the benefit is likely to be greater. The risk of COVID-19 infection is higher, and the benefit of the vaccine is likely to be greater. Any risk from the vaccine is purely theoretical now. It's a very safe vaccine.
Yeah. Well, let's move to another special population of sorts, and that is the 20 million of our friends and neighbors who have had COVID. And the studies, as I understand it, excluded people if they had known COVID disease, but a lot of people had antibody, not a lot, 2% or so. What can you tell me about recommendations for people who have had COVID? What's the safety profile look like, and what's the recommendation for when the timing of the vaccination?
Yeah, so several hundred people did get into the trials. They didn't have known COVID. They were probably those folks that couldn't get tested back in the spring. Testing wasn't available, or they had asymptomatic infection, which CDC says maybe 40% of infections are asymptomatic. And they were enrolled, vaccinated. Later, nucleocapsid, nucleoprotein antibodies were looked for, and that's how they defined people that had pre-existing infection that they didn't know about. And those folks did fine in terms of safety. There was no difference. And in terms of efficacy, there was an efficacy signal that was similar in those folks. So the guidance is that people that have previously had COVID should take the vaccine. There's no reason not to. When to do it is another question we're getting a lot, Mike, and it really depends on when the COVID was. If it's in the remote past, go ahead and get it. There's some thought that in your first few months after COVID, you'll have a level of immunity that maybe you could, maybe this is altruistic, or maybe you'll get a better take if you wait a little bit longer. Certainly, one clear guidance is if you have acute COVID and are still symptomatic, you shouldn't take the vaccine. What I've been saying is that, yeah, you could wait up to 3 months, as has been suggested, but if you don't want to wait that long, I think waiting a month or so post-symptom resolution is fine. And then you'll get a nice boost, I believe. And whatever our concern is, and we're still learning for waning immunity, it's clear you can have a second infection. But immunity is going to wane eventually based on what we know from other coronaviruses. Then we take that out of the picture.
Yeah. Well, speaking as an N of one, as everyone I think knows, that I had COVID back in March of 2020, and I got my vaccination about 2.5 weeks ago. And my reaction was basically like having COVID over again, not the respiratory part, but the fever, muscle aches, chills, all that. So I'm pretty confident my immune system recognized this guy and reacted. It only lasted a day. It was miserable, but it wasn't horrible.
Yeah.
And it just has gone away, and I feel fine. The question for me is, should I get that second booster shot? I'm kind of thinking maybe postpone for a little while. I think I've got pretty good immunity, but we need more data on this. And I do have blood that's been drawn to sort of look at my antibody levels before and
Yeah.
after.
That's actually another way we might be able to save vaccine. If we can get these data that show, yeah, if you've had it before, you only need a single shot,
That's
you'll get
it.
a
Yeah.
nice, yeah. So
Yeah.
I like that.
Yeah. Well, one of the things also that we're talking about that, you don't have a crystal ball anymore, I guess, than anybody, but what's your best guess about the longevity in terms of the
Yeah.
slope of the immune response that you've seen, at least in the first 2 or 3 months? When
Right.
might people need another booster?
Yeah. There was a nice small like letter paper in the New England Journal 2, 3 weeks ago for Moderna showing from their phase 1 participants. So now we're far enough out to have 3 months of neutralizing antibody data post second vaccination. And at 3 months, it's still very robust. There's a little drift down. Just looking at that, and of course, not knowing, well, where's the threshold? Where do you cross the line where you're not protected? And again, circulating antibodies is not the whole story. Memory T cells, memory B cells will kick in as well. But my guess is that we're going to be out close to a year, would be, if not longer. Just looking at that, I think that we don't know the correlative protection yet, so this is all just hand waving. But it looks to me like 3 months after 2 doses of the vaccine, we've still got a high level of neutralizing antibody.
Yeah, I think you're right. It's going to be longer than I think. It might hopefully it'll be long enough so that the herd immunity can kick in and we'll start to get rid of this guy, and so we don't have to deal with him anymore. That would be good. Speaking of the sort of mechanism and how it might relate to epidemiology is, it seems like what happens with this vaccine, assuming somebody's never had COVID before, is that they get an immune response that should they get exposed to the virus down the road, they certainly don't get symptomatic disease, or at least not much of any symptomatic disease. But the question is, are they transiently infected? Probably so. And during that time, is there any evidence of shed where they might be at risk to another person who hasn't been vaccinated yet, who's not had COVID?
Right. Yeah. So if we sketch the whole spectrum, profound protection against severe disease. And then 95, I mean, I'm talking over 95%, it's impressive. And then against symptomatic laboratory confirmed COVID, 95% protection, very strong. And we will get the answer to the asymptomatic infection, which is what you're talking about. Somebody who might be infected and able to spread the virus. Would that happen? Does this vaccine protect against carriage or asymptomatic infection? And we just don't have that answer yet. Again, with those nucleocapsid antibodies, we can look for people in the trial who were negative to nucleoprotein antibodies at the beginning and then have it later, because the nucleoprotein's not in the vaccine. So if they develop antibodies to that during the trial, they got infected. And if they never reported any symptoms, that's asymptomatic infection. And so then we hope again to see a lot more of that in the placebo group than we see in the vaccine group. And those data somebody probably knows that right now, Mike, because they've got a lot of data to go through. But I think we'll hear about that. My guess would be sometime relatively soon, I hope.
Yeah, I would think so too. Another thing in the news the last 2 to 3 weeks has been this variant that was originally described in London and South Africa, recently in Colorado, California, in the US, probably everywhere, I would guess by now, that's more infectious than the parent wild type virus. But according to the data I've read, that these mutations, which are pretty well characterized, do not sort of overlap with the antibody produced by the vaccine, and therefore the vaccine should still work against that variant. Is that accurate?
Yeah, the way I like to talk about this is that these guys, like HIV, they're RNA viruses, and they're professionals at mutating. And with one of the RNA vaccines, they looked at 15 of the variants. This was in a paper published in Nature, and all 15 were covered equally well to the canonical strain, the original strain from Washington State in the U.S. So all the variants that had been studied in that paper were covered. My guess is, and I think most people feel this is probably true here as well, that the vaccine will cover these variants. There's no evidence that the UK variant is causing worse disease. We will have some answers soon about vaccine protection against it. The evidence for increased transmissibility is based on modeling. And I think I'm beginning to hear some whispers from a few different labs that that they're seeing some biological evidence of more rapid spread or more avidity binding the ACE2 molecule, the viral receptor. I think we're going to see some of the biological explanation for this purported and probably true increased transmissibility. Still a possibility if none of that were true, that it's just human behavior leading to more spread of a variant that just has sort of gotten seeded, and now with cold weather and gathering for holidays. But I think most likely, based on what I'm hearing, that there's something to it. But masks and everything else we're supposed to be doing will be equally effective against
Yeah.
this.
Yeah, that's exactly right. And, if we put this whole conversation together, and I'm about ready to move into the QA because we've got a fair number of questions, over 30 that have built up, is that even when you get the vaccine, you still want to wear a mask, you still want to maintain all the rules. You will have, I'll speak from firsthand, after having COVID originally in March, by April, May, when I was going out and seeing patients again, I did kind of feel like I was a little bit protected. I still use PPE, but it was different, I think, than some of my colleagues. So I think the vaccine recipients were going to feel the same way, but really important to continue that, right?
Yeah, that's what yeah, absolutely. We've got to keep wearing the masks, social distancing, hand washing. What I'm hearing from people is that same thing, just a little bit of a personal security feeling, but they're not going to throw away the mask. What we need to do is get to herd immunity, and nobody knows what that number is. We're still not really sure what the R naught is for this virus, but we thought it was maybe 3 or 4. Now maybe it's 5 or 6. So we were talking about 75% level of herd immunity. Now there's some talk about 85 or even a little higher, and which would mean we got to go a little deeper and further with the vaccine. So when we get to that, then the pandemic stops.
Right.
And
So for some of the folks who haven't kept up, it R naught means the probability of one infection infecting another person or two, and you want that value less than 1 to stop an epidemic. And the higher it is above 1, then that just means it's much more infectious, and more people,
right.
once it gets transmitted, it's like a sort of a nuclear reactor approach where it just keeps
Yeah.
exploding.
The world champion is measles.
Yeah.
12 to 18 secondary cases for each primary case in a fully susceptible population. And their herd immunity requires over 95% of people vaccinated. The
Yeah.
formula is 1 minus 1 over R naught. And our guess here is somewhere between 75 and 80, 85. We'll see.
Yeah, and so I'll finish up my questioning. We'll go to the questions with this point that, I think a lot of us, to get to that herd immunity, a lot of us are concerned about the acceptability of the vaccine. And what are your thoughts on that in terms of the uptake?
Well, it's uptake, it's also the distribution and allocation. So vaccines don't save lives, vaccinations do. That's my friend Walt Orenstein, who's one of my heroes, and it's so true. So we got to get these things into people's arms. So it's the distribution, but then it's also trust, particularly the communities most affected by COVID, where there are many reasons for historical and ongoing, continuing distrust of government, of science, based on some things that have been done and maybe a lack of participation. I am glad that up to 40% of participants are minorities in the trials. So that's a good thing. And we can go to the communities and say, hey, this has been studied. It's equally safe and protective in Hispanics and African Americans based on the data in the trials. But we've got to invest in these communities, because we're going to need participation from the communities most affected, the essential workers, the bus drivers, all the folks that are most at risk. And so they have participated in the trials. We've got to get the word out, and equally important job to the distribution is the education piece now. And I just would say to the, I don't have my glasses, to the 399 people, I would like to ask you all to be vaccine ambassadors, and I think you probably already are, and get the word out about the safety and the importance of achieving herd immunity.
Yeah, it's great. And I've noticed just anecdotally in the last 3 weeks, even as the vaccine is rolled out, I've seen a shift in the desire of a lot of people saying, I'm not going to get it, or I'll wait and see. Well, now that it's rolling out, and especially so many healthcare workers who are in the know about the science, a lot of the national leaders have rolled up their sleeve literally. And now I see my phone's blowing up. I'm sure yours is as well. Like, how can I get it? When can I get it? My family member XYZ. So I think that's it. So let's move on to questions. The first couple of the original questions we've already addressed in terms of differences between vaccines. But this is from Elizabeth Sherman. It said, just basically saying HIV patients are asking her, should they get the vaccine? I think you've addressed this, but seems like the answer is yes, right?
Yeah, I think there's no reason to think there'll be more of a safety problem. It's a very safe vaccine. We don't have efficacy evidence because the numbers were small, but in a few hundred people in the phase 3 trials, safe, tolerated, no reason not to have a discussion and consider getting it. We don't know if they'll respond as well if they have deeper immunosuppression, but particularly those that are well controlled, we would expect that maybe like many other vaccines, they'll get a response.
Yeah. This is from Soljanya Kenyuri. Are you seeing neurological effects? We talked about the transverse myelitis. There's been some Bell's palsy, but it seemed to be balanced between the groups, maybe a little bit more. And then the notion of somebody who's had Guillain-Barré, should they get the vaccine? What do you think?
Right. Yeah. So there's not been any Guillain-Barré seen with any of the vaccines, so that's good news. And in fact, in general, Guillain-Barré is not considered as either a contraindication or a precaution to any vaccine. Same thing here. So I think that that's a decision they can make. Some people that have had it are a little bit nervous about getting vaccines just because of some of the things that are out there. But I think it can be done. We're not seeing other neurologic problems, that are any different. Mike, you mentioned the Bell's palsy. What FDA and CDC have said is it seems to be roughly at about the background level in the population in general. And then we've already addressed the transverse myelitis with AZ, but really very clean safety data. Yeah, just one, and that's not clearly linked.
So Jonathan Rosenthal asks, what about potential unforeseen side effects from the new mRNA vaccine? For example, emergence of autoimmune disease that might be triggered by this and that type of thing.
Well, Jonathan's question is great because the trials for Moderna and Pfizer combined 75,000. Now we've got over 3 million, people in the US alone. So we're going to learn some things. This is a time for humility, modesty. I mean, I hate to say it, but COVID has defeated us repeatedly. And I think we need to be humble with the vaccines as well and say, we'll probably learn a few new things. We always do when you roll out from clinical trial where we study efficacy to the community where we study effectiveness. And, that I think that's what I'd have to say. We're going to learn a few things. He asked a specific, I forgot what it was, Mike.
Well, it was just about, will there be autoimmune diseases?
I wanted to just say, so immunosuppressed participants immunosuppressed individuals were not allowed in the trials, but autoimmune conditions were, and we did not see any. So if you had an autoimmune condition, but you weren't on immunosuppressives, you could be in the trial. There were several hundred enrolled. There was no, one of the concerns about adjuvants and self-adjuvanting vaccines might be, are you going to trigger an autoimmune, turn on the immune response against whatever these cross-reactive molecular mimicry epitopes are? Nothing like that was seen with this relatively small data set. So Jonathan, I would say stay tuned, but I don't expect that we're going to see autoimmunity.
Yeah. Nadia Kurishi asks, how many times will the spike protein be made before the mRNA gets? Do you have any sense of the burst size of spike protein?
I wish I had an answer for that. I don't really. The way I think of it is that the the lipid particle goes into the cell, the RNA is released, you start making, and then pretty quickly RNA is degraded. We know RNA is very sensitive and it gets degraded quickly. The total amount of protein made, remember it's a spike trimer, it goes to the plasma membrane and stays there. My guess is it's a few micrograms at most. It might actually be nanogram level, but because you've got such a strong adjuvanting effect going on, you're getting a really, really good response. So remember, a flu shot, each of the 3 components are 15 micrograms each. I think this is just a fraction of that being made, but because it's a strong response. And again, the RNA probably is made for a matter of hours or a day, or so, makes the protein just for hours or a day or so, and then you're done. But I can't put a number on it.
Yeah, I think the backhanded way we know that the mRNA doesn't hang around is all the extremes we have to go to ship the thing, right? The amount is 80. That's because it'll degrade and won't be available for use.
Which is one of the reasons the AZ vaccine is so important, because it can go in a fridge and it's also very cheap. So it's a vaccine for the whole world that will be easier, just as an aside.
Yeah. James Sunstrom asks, why is a stabilization with proline needed?
Yeah, we want to lock in the pre-fusion configuration. So after the virus attaches with its spike protein to the ACE2 molecule, the conformation changes. You don't want to make antibodies to the spike post attachment, or if it somehow triggers and goes into the post-fusion, post-attachment configuration. So we lock it into the circulating free virion particle configuration with those 2 prolines. And you induce the immune response to that. Now you're ready to go attack free virus.
Okay. Lorraine Tassilo asks, is there a diagram to explain how mRNA attachment and entry, maybe you just use your hands, cell, hand,
yeah, yeah, I do think, well, I think of it as linear, actually. So you have RNA, that's the message, and you go into the cell and you tell the cell, make the protein. So RNA, protein, and then the protein triggers an immune response, then immune response, that's the 3rd thing. And then 4th thing, protection. So message through the RNA, make the vaccine spike protein in the body, make the immune response, and then protection. 1, 2, 3, 4. And if you want to take it to the adenovirus vectors, back up just 1 step. Those are DNA viruses. You've got the spike DNA snuck into this non-replicating adenovirus genome. So you go DNA, RNA, protein, immune response, protection. You have 1 extra step.
And that's regular transcription, and that takes place in the cytoplasm. Again, it doesn't seem to need to go to the nucleus to make all that happen.
Right. No danger of integration. The RNA never makes it into the nucleus, and we don't have any reason to feel that this is going to get reverse transcribed into DNA.
Gabe Teferi asks about polyethylene glycol. Is that in the vaccine, and what does it do?
Yeah, so there are at least 4 different lipids and some other chemicals in the lipid nanoparticles. So yeah, the vaccine's got the RNA and then this lipid coating, and there is polyethylene glycol there, and polysorbate is something that is somehow related. So this question has to do with the allergic reactions, Mike. If you have a known anaphylactic reaction, this is a contraindication, a known anaphylactic reaction to polyethylene glycol, PEG, which many people will know, or to polysorbate, or to a first dose of a COVID RNA vaccine, you should not take the COVID vaccine. These are very rare. With regard to the anaphylaxis, people with anaphylaxis can take the vaccine. Maybe bring your EpiPen, and of course we give the vaccine in secure places where people have epinephrine, where they know how to handle airways. We watch people with a history of anaphylaxis longer, 30 minutes rather than 15. Remember 3 million doses now in the US, and we've had a handful, but this is really important. Vaccinations in the US, about 1 in a million or a few in a million result in anaphylaxis. All vaccinations. It just happens. It's a known complication of vaccination. I'm not convinced this is anything different than what we see with all the other vaccines. We've seen a few, and we've given 3 million doses now.
Yeah. I think of Peg as a song by Steely Dan on the Age album. I'm just saying. Marguerite Owens wants to know, is there any studies that include end-stage renal disease?
Yeah, no, unfortunately, we didn't. That was, we excluded people with severe chronic disease, mild or moderate, well-controlled chronic disease. And so that was an exclusion. I think that needs to be done, but it hasn't been done yet.
Joe Cowper's asking about Moderna dose being halved for the younger populations because their immune system response is going to be stronger. Is that something you think we might be moving forward with to get more vaccine available?
There is some talk for children about studying a half dose. There are pediatric trials underway. Remember, the Pfizer vaccine's has an EUA for certain children, 16 and 17-year-olds. It's being studied in 12 to 15, and trials as young as 5 are planned for all of the vaccines. I think if this question had to do with adult half dosing, it may be coming in part from AstraZeneca, but also that there is a pretty decent immune response even to lower doses of the RNA in younger adults. That's true, and maybe I think maybe that's the question. Older adults, you had to use the doses that were licensed, but right now that's not being planned. That's complicated. Listen, when you're talking about vaccinating 330 Americans, if that many want the vaccine, the more curly cues you put into the plan, you got to make this simple.
Yeah,
So
I
it's
agree.
something that could be addressed, but it might be too complicated.
Sarah Miller's asking about those over the age of 65, and when you slice and dice the data, Pfizer seemed to have 93% efficacy in that population, Moderna 86. Is there a difference? Does it matter?
Yeah, I don't think we should look at it that way. My view is that we had pretty good participation from older adults, about a quarter, and both vaccines were highly effective. There's a little degradation. The immune responses, if you look, the neutralizing antibodies, they're a little bit less, but both vaccines are highly effective in older adults. Mike, what I'm telling people is take whatever you can get, because you don't know, with these supply problems,
Yeah.
let's don't be picky. They're both 90+%, even in, or thereabouts, even in older adults.
Well, that gets to a question by Richard Halbrick, who's asking about if you get an mRNA vaccine first, can you follow it up with AstraZeneca second, or Pfizer versus Moderna?
Yeah.
I'd recommend it, but what do you think?
Yeah, not recommended. You're right. The U.S. guidelines are to take the same vaccine. Here we have just Pfizer and Moderna. So if you started, just because it hasn't been studied, and that's a standard recommendation from the health agencies. Sometimes these things have been studied. People move around. They may live in an area where there's Moderna, then they move, and now there's Pfizer. So maybe it's going to happen some, and it might eventually be shown to be okay. We've studied this actually with certain pediatric vaccines, where you took the HPV vaccine from one group and then you tried it from the other group, and it turned out pretty well there. The half, yeah, I'll leave it there.
Yeah. Okay. So, and you're right. That's another curly Q. I like that term. Questions are really great here. Another one from Melissa Weiner, asking about uncontrolled HIV patients. I think what we want to do is get them on antiretroviral therapy, but assuming we're trying to protect them against COVID, no data on that.
Yeah, no, they weren't studied. The recommendation there would be that they're immunosuppressed. You can have the discussion. It's not contraindicated, likely to be safe. We don't have the evidence to support safety and efficacy, but certainly can be done. But it does give me a chance again to make a plea for cocooning, herd immunity, the family around people that are, in all likelihood, not as likely to respond quite as well because of their immunosuppression. Really important that everybody in the family, everybody around them, get the vaccine when they can. I know that probably many of those family members can't get it right now, but this is what we talk about with pertussis, for example. When babies are under 6 months, there's no way they can get protected from vaccines, and so we got to vaccinate the whole family.
Herd vaccination of the local herd.
Cocooning.
Yeah, exactly. So Peter Kadlecik has a question that I was going to ask but ran out of time, and that is the ethical considerations of continuing, for example, the Pfizer study. The data are known, yet the placebo group continues for a while. What are your thoughts on that?
Yeah, this is something that is taking up a lot of my day right now. We're unblinding people in both the Pfizer and the Astrazeneca trial who would like to be unblinded, who are being offered the vaccine. So a lot of doctors and nurses at my institution were in these trials. They're now being offered to get one of the vaccines, and they need to know. The trials have been amended to allow us to do this, and we're doing it. So we're doing the right thing for our current participants. What is also going to become very important will be what about people that aren't in the first several levels? They're not 1A, 1B, 1C. Can they be unblinded? And just recently, Pfizer has switched and now is going to use the same approach that Moderna is, that vaccine will be made available within the next couple of months to pretty much everybody in the trial. So who would like, some people may choose to stay blinded. I've had one person tell me they wanted to stay blinded. Of course, that person had very strong reactions to both shots. So I think that individual knows what they got. The trickier thing, I think all of that is kind of a no-brainer. You got to do the right thing and take care of people. Admittedly, we're going to lose our ability for long-term, a lot of our ability for long-term safety and efficacy, but you can't withhold from people that need to be protected now that we know it protects. What'll probably happen with future trials, like we didn't get to this yet, and maybe we're going to, the protein platforms. So the Sanofi trial that will be starting in February with a lot of other sites, it may well be that design is going to have to include not a placebo comparison group, but actually an active vaccine.
Yeah, non-inferiority.
So the designs are going to probably have to change.
I think it will for sure. We have about 10 minutes left, so we'll make quick questions and answers here. Thomas Motto asks about solid organ transplant recipients.
Yeah, not studied. They were excluded, and trials will be coming for many of these immunosuppressed groups, pregnant women, children that we didn't get to in the main trials. Unfortunately, the way we do the big initial phase 3s are usually adults who are not immunosuppressed. We want to make sure that it's safe and effective. Then we go to the other groups. So this will be studied. I know there are transplant studies being
yeah, the problem is we don't have time, right? Because these folks are vulnerable, and
right.
So my inclination is to vaccinate them.
Yeah.
As an at-risk population. Tom Dietz asked about monoclonal antibody. I'm in my scrubs because I was just at our monoclonal antibody clinic about an hour and a half ago. The timing of the vaccination after someone had COVID and got early monoclonal, how long? Yeah. Again, 60, 90 days?
Yeah, half-life is about 20 days for these monoclonals. So you want to get out 3-4 half-life. So we're talking just what you said. The official guidance document from CDC says 90 days.
Yeah. What about comparators of immunity from COVID versus immunity from the vaccine in terms of longevity? I've heard that the antibody response tends to wane a little faster in those with natural infection.
Yeah, so people that were naturally infected and not vaccinated, we know there's waning of immunity. We know secondary infections can occur, but it's pretty unusual. It can happen, but it's certainly not common. Remember, we've got 10 million people infected now, and we haven't seen a million second infections. It's going to be pretty unusual, I think, rare to uncommon, but it does happen. So there is waning, probably over 3 months. CDC likes to say that less than 3 months, you really don't see reinfections. That's why it's reasonable to consider waiting out to 3 months after natural infection to get your vaccine. You can get it sooner if you want. I think the duration for the vaccine, as we talked about a minute ago, Mike, is likely to be 9 to 12 months, if not longer. So I think we're going to do better with these vaccines just based on what I'm seeing. 3 months out right now with again, as I said, the Moderna vaccine, there's very little drift, a little bit, but not much.
So Sylvia Stamperns asks about the very old, if you will, the folks over the age of 85 or 90, who I'm pushing to the front of the line once we get into phase 1C or whatever it is. Is there a preferred vaccine in that age group, or is there a difference in data?
Yeah, I really don't believe there is, Mike. I think they're both highly effective, and I agree with you that, in fact, we know now that 1B is going to include 75 and older. These older, my mom's 88. I'm just dying for her to get the vaccine. I told her, whichever one you can get. Again, take what you can get. You may not, it's not, most of us are not going to have a menu option to pick, I
Yeah,
think. So
yeah,
they're both highly effective for this group.
yeah, I agree. Paul Aaron's asked the question. I think we've already addressed it somewhat about the prime boost in a smaller dose. I think we kind of addressed that. Jenny Town is asking about what turns off the body's replicating the mRNA. Where does it go?
Oh, yeah. So RNA is very fragile and is easily degraded. Natural process in the body. We make messenger RNA when something's happening, but we don't want to keep making that protein for a long time. It gets shut down and degraded, enzymatic degradation, other pathways. So that's a normal body process. It goes away within hours to a day or two.
Yeah, I learned that very early on in my lab experience in the 80s, where we were doing mRNA experiments, and just a little bit of RNAs destroyed the experiment. So it's very fragile. Um, let's see. Dwight Ferris is asking about how to deal with the placebo arm, and we talked about that a little bit.
Yeah.
Um, uh, we talked about half dosing. Let's see what else we got here.
Let me throw
Yeah.
in while you're looking about the AstraZeneca, a lot of the half dosing comes from AZ. So there was a report that giving a low dose prime, the half dose prime, and then the full dose boost was great, gave greater protection. And that's published in Lancet. But there's a problem, and that is that the interval also was very different. They had intervals ranging from 4 weeks to 12 weeks. And we know in general with vaccines, the longer the rest between prime and boost, the stronger the response to the boost. So they varied not only the low dose prime, but they also had longer intervals. It's very hard to sort out which of was more important there. I think we need more data.
Yeah. Siobhan Solano is asking about, this is a tough one, multiple myeloma bone marrow transplant who received CAR T cells.
Oh, yeah.
I bet that was a large group in the studies.
Yeah, unfortunately, of course, not studied. But again, not a replicating vector vaccine,
Not
not a
much
live
risk.
viral vaccine, likely to be safe and well tolerated. It's are they going to respond? Well, I mean, the transplant team would know best. There's no reason to not give it, from my point of view. If you think, and we know they're at high risk for severe disease, so the upside is probably good, the downside is probably low. Makes sense to have a discussion.
Yeah, Joel Ong is asking about is in a private practice, and a lot of the staff are scared and don't want to take the vaccine. And what should you see? I'll tell you what I've done, and I think it's worked. And that is just compare the world in two scenarios. One, COVID continues like it is, and there's no vaccine. When does it end? How many more people die? Versus COVID continuing, but then a vaccine and herd immunity happens. And this gets to a question that I saw earlier, and I don't have the name associated with it, but how long will it take in your estimation? But let's take out let's take on that second scenario. My best guess is maybe if this all rolls out and does well, certainly by maybe Thanksgiving, Christmas of next year, we can start thinking about a return towards normal. But if we didn't have vaccines, next Thanksgiving and Christmas would look at least as bad as what we have now, not to mention
Yeah.
that almost 1 million people probably would have died by then.
Yeah.
And so when you weigh it out, this is a real holiday miracle, whichever religious background you may have. It's a miracle in the miracle season. And I think we should really embrace that, right?
Oh, absolutely. I agree with all that. And I also think that, one, I'd love to be able to talk with that group of staff and just have a discussion, find out what's on your mind. What are you thinking? What are you concerned about? And then also to say, you may feel like you don't need protection because you think, you're young and healthy, and we know younger people do better. But as an altruistic act, and for your patients who are at high risk of death or severe complications, that's
It's
a
7
reason to get
o'clock.
vaccinated, to protect those vulnerable people that that you're caring for. We're not going to mandate this vaccine. It's not a licensed vaccine. It's got this emergency use authorization. But I do think, particularly for those of us in healthcare, there's a bit of a social contract there that we really should think about to protect our patients.
Okay, we've hit the top of the hour. We still have remarkably almost everybody who started with us, so I guess that's a backhanded compliment. I'll just finish with two questions. We'll go a little bit over.
Sure.
One is the notion of, is it worth checking antibody afterwards, especially in somebody who didn't have any reaction at all to the vaccine? Is it, and if you would check antibody, which assay?
Yeah. No, it's not worth checking. A 95% protective vaccine does not need a post vaccination test for immunogenicity. We know this is immunogenic. If you were going to do it, this comes up a lot in our trials, people wanting to unblind themselves, go out and get a test. But the problem is some of the tests like Roche and Abbott measure nucleoprotein. They don't measure spike antibody. They measure antibody. So you can't get just any test. It would have to be one that measures the spike directed antibodies. And some of the tests don't even tell you what antibody. It's proprietary. They don't tell you what the antigen is in their test. So it's a little bit tricky business. I don't recommend it, Mike. You don't need to do that.
I agree. And I'm going to finish with a question from our colleague, Victoria Johnson, who we both know very well. And just asking about her 91-year-old mom. We talked about this, but do older folks, I'll just reiterate the question, have the same 95% efficacy, do you think?
I think it's very high, and probably not quite that. Remember, 95 is across all the age groups, and we do see a little drift down in the older groups, but it's still very, very high, in the range of probably high 80s, something like that. So highly protective. And this is remarkable about these RNA vaccines. In fact, the antibody levels for the Moderna vaccine were as high and even higher in the older adults. It was striking in the phase 1-2 data. So Vicki, my mom's 88, your mom, hopefully they'll both get the vaccine soon, and I expect they'll have a very high level of protection.
That's a very optimistic note to end on. Mark, this has been joyful, and the time went by very fast, as we thought it would. Lot of information that I think is spot on and very much needed now. And maybe so many questions are still needing to be asked. Maybe in several months, we might repeat this with updates where we can answer some of the unknowns. Thanks to everybody, especially the IAS-USA staff and all the audience, for hanging in there with us for the last hour. A couple of key points. This dialogue will be available on the IAS-USA website. And it will be, if you download it or you watch it online, there will be the opportunity for CME. This has been, as I said, just a great hour that I think we can say that there's not only this dialogue, I think that's very informative. There's a series of them that have been on, ranging from epidemiology to populations to basic science, that are there for anyone who would like to go scope it out on the www.iasusa.org website. We will conclude here. Mark, thanks again for a great job, and always fun connecting with you, even if it's a longer distance than Atlanta going to New York.
It was a great pleasure, Mike. Thank you.
Okay, we'll sign off here. Thanks again, everybody.